Okay, so it's been 20 minutes here, so what we're going to do is grab the samples. We hit stop, and that'll stop. To add to it, I didn't mention it in the previous step. At this point, I usually label the samples of what they were. So these are like wild type cells, for example, different UV doses, rather than numbering 1 through 12. At this point, I typically label the tubes. Okay, so next step is we're going to start to precipitate the DNA. So the first thing is we're going to add glycogen, which helps to co-precipitates with DNA. So sometimes it helps you see a pellet of DNA. So again, I already made the dilution. 14 microliters of glycogen into 350 microliters of PBS, and now I'm going to add 25 microliters per sample. Okay, so that's done. Next thing is we're going to add is 1.1 volume of sodium acetate. So it's 1/10 the volume of the liquid. Based on my prior experience, we started with 1 ml of cell culture media. Due to evaporation and all the centrifugation steps, it tends to go down to about 900 microliters or so. If you want to validate that, you can by just using a pipettement and pulling up and see what the volume is. So I'm just going to assume it's 900 microliters, since we need 0.1 volume, which is 90 microliters. So I'm going to grab my P200. Add 900 microliters of sodium acetate. And this is pH 5.5. So notice when we look at the samples, the color will change to yellow. So for example, this is red. We add this, it's going to go yellow, lowering the pH. So it makes it slightly acidic, and again, DNA is an acid, so it'll precipitate under acidic conditions. Okay. And so the next step is we want to add isopropanol. So, let's see if I have more over here, yeah. We're at 0.7 volumes of, so we started with, again, 900 microliters, 0.7 volumes is 630 microliters. And if the volumes are slightly more or less than 900 microliters, you can readjust things. If your volume is less than 900 microliters, you can bring it up with something like water or TE or PBS. And the precipitation will be optimal under the right conditions. And so if the volume is off, it changes the concentration of things. This will make the tubes fill up almost nearly to the top. So it's everything should fit. Tubes. Either vortex, or what I often do is just kind of invert it like this. Make sure things look like they're mixed up well. And then what we'll do is we're going to centrifuge for 30 minutes. Okay, so we're going to spin at maximum speed, or 20,000 G, it's almost maximum speed, for 30 minutes at 4 degrees. And when we're done, what we're going to do is we're going to pour off the isopropanol. So this centrifugation under these conditions, and with the salt and the sodium acetate and the acidic conditions and the alcohol, causes DNA to precipitate. So it'll be down at the bottom of the tube. So the supernatant will just have the alcohol that we can pour off. And then what we'll do is wash once with ethanol, and then it'll be all done.